And so talking more about liquid biopsy, there are three sections or three types within liquid biopsy as well. One of them is CTCs, which is whole intact cells, which is circulating tumor cells, as you can see over here as well. This is a tumor when it's spread, or even if it is not spreading already in early stage disease, sometimes this tumor starts shredding their cells inside the blood vessel. And this is what we are trying to evaluate and assess for different biomarker testing. So one of them is the CTCs, which is just means that circulating tumor cells. It's as simple as it sounds that these tumor cells, when they're circulating within the blood, they're caught and then analyzed further. The second one is ctDNA, or it is more broadly called cell-free DNA. So cell-free DNA, and you can look over here. These are the pool of DNA fragments from cell death. Within this cell-free DNA, it just means that it's not in the cells, it's free of cell, the DNA. Within this, the circulating tumor DNA is the specific tumor-derived fraction used to identify genetic mutations. So that's the second entity. And the third one is RNAs. So the RNAs refer to tumor-specific mRNA transcripts, just particularly mentioning that there are different types of RNAs that provide, again, real-time measure of gene expression levels rather than just structural DNA changes. More so, in a broadly speaking, these are DNAs and RNAs, and when you analyze sometimes in combination, it can give you more data of genomic testing. So when you are discussing with different doctors, like these are three different entities that can be studied. Very often right now, circulating tumor DNAs are studied in liquid biopsy, but there is more and more data as well as more and more platforms or industry who can do circulating tumor RNAs as well. Just again, a little bit of difference. They're whole cells, DNA fragments versus RNA transcripts, and focusing more on key insights that it can give cellular structure, viability, and multiparametric analysis, whereas circulating tumor DNA could give genomic alterations and tumor burden, and circulating tumor RNAs can give dynamic gene activity and marker tracking. You can read this further in one of the study and one of the review article that we had published, which is available over here for your reference as well. Going into liquid biopsy utilization, again, we are talking about lung cancer, but I'm giving a reference of prostate cancer over here in this figure because it's a very nicely prepared figure, which gives a very nice information on how liquid biopsy can be helpful. So first and foremost, it can be helpful in treatment guidance. So various studies in different kind of solid tumors as well. It has shown that depending on the ctDNA approach or ctDNA analysis, it can help us decide whether adjuvant treatment can be given or what kind of treatment can be given. And sometimes it can also help us in neoadjuvant treatment as well. Neoadjuvant and adjuvant difference is just simple, that neoadjuvant is before surgery, whereas adjuvant is after surgery. At the same time, it can help us in treatment response. So it means that whenever treatment is given, and if we want to see whether the treatment has worked well or how the cancer is progressing or if the cancer is coming back, it can help us analyzing in that as well. And similarly, in molecular or minimal residual disease detection, which just means that how much disease is still there in the body at the lowest possible level. And it just gives us a longitudinal surveillance of ctDNA as well after surgery, with sometimes really high sensitivity and specificity as well. It can also give us data of treatment-resistant characterization, which means when we look at this tissue, liquid biopsy, it can give us more information on how the tumor is evolving further as well. Just to see how this landscape is evolving constantly, there are multiple different companies, and some of them I might would have missed on this slide as well. But these are some of the major companies that are constantly being used for liquid biopsy or for tissue biopsy and so on and so forth. Another one over here could be Foundation as well, apart from all the ones that are already mentioned in this slide.